Okay, so guys, today I will be just giving you a short description of the CRISPR technology, or in other words, we can also call it gene editing, right? CRISPR technology or the gene editing. So right in front of you, you can see that in the keywords box, the description of gene editing is given to you, which is a form of genetic engineering in which the genome of an organism can be changed either by deleting, inserting, or replacing a length of DNA using a method such as the CRISPR or Cas9 system. So what are we doing by gene editing? We will be using, it's of course a way of genetically engineering something in which we are changing the genome of any organism, and that organism can be as small as a fruit fly or as big as a human being, and we are changing the genome by deleting, inserting, or replacing a length of DNA. Of course, when we are doing it on different organisms, our aims and objectives will be different. For example, if we are doing this on tiny insects or tiny organisms like plasmodium and bacteria and viruses, so maybe we are doing some research work, or we are designing a drug, or we are designing a vaccine. And if we are doing it in human beings, then the objectives will be different. If we want to treat a genetically transmitted disorder, right? An autosomal recessive disorder, let's say cystic fibrosis or any other disorder, by means of deleting, inserting, or replacing a length of DNA. Now, if you remember, guys, when I was doing gene therapy with you, I had clearly told you about the definition of gene therapy, which was that gene therapy means you are introducing a normal functioning copy of a dominant allele into a person's genome, right? And gene therapy is only applicable in diseases which are autosomal recessive. Okay, if a person is homozygous recessive for that particular disorder, he will have symptoms. So if we could treat that disease, let's say a disease like cystic fibrosis, which is autosomal recessive, which is following the autosomal recessive pattern of inheritance, I can cure that by gene therapy by introducing a normal functioning copy of the dominant allele. And in this way, the symptoms will be better. And when I was giving you the definition of gene therapy back then, I had clearly told you that never write down in gene therapy that you are replacing a faulty allele with a healthy allele. Don't do that. But here, when we are doing gene editing, you can use this definition because gene therapy is completely different from gene editing. In gene editing, yes, you are deleting, inserting, or replacing a length of DNA using a method such as the CRISPR or Cas9 system. Okay, now before I give you the description, I would like to tell you that in your A-level syllabus, in your exam syllabus, I would not consider this to be a very major part of your syllabus, because there's a very brief learning outcome which expects you to know the basic layout of how this procedure is performed, right? Not going into or without going into too many details. So I don't have right now in front of me on the screen the soft copy of the syllabus, but I would advise all of you to go through the soft copy of the syllabus and the scheme of work to see how much you need to know about this. The bottom line is that you need to know about this very briefly, not going in too much detail, right? How many pages is this of your textbook? Is this topic can be seen in your textbook? It starts from this previous page in which you have the definition of gene editing, right? And then you have two more pages. As you can see, this page in which it's showing you what are the steps of CRISPR, right? I'll just zoom into it. These are the various steps which I will be describing today, and some clinical applications. That is it. It's just 2 pages. Before I start describing this, I will share an article with you, and I'm going to read from that article. It's an article from the Harvard University, and it's about, the topic is CRISPR, a game-changing genetic engineering technique. So let's first read this article, which will make it easier for you all to understand your textbook content of the same topic, right? So it says, of course, it's a game-changing genetic engineering technique. It has revolutionized the scientific community. Let's see what this is, right? Number one, CRISPR stands for, it's an acronym for clustered, regularly interspaced, short palindromic repeat, right? This name refers to the unique organization of short, partially palindromic, you all know what a palindrome is, repeated DNA sequences found in the genomes of bacteria and other organisms. So these are repetitive palindromic DNA sequences which are normally or naturally found in the genomes of bacteria, specifically bacteria and other microorganisms when they are attacked by bacteriophages or viruses. What is the benefit of these? CRISPR sequences are a crucial component of the immune system of such simple organisms, meaning that these repetitive DNA palindromic sequences are found in the genomes of bacteria, especially when they are invaded by viruses. Bacteria develop them naturally as a part of their immune system so as to protect themselves from the viruses. I'm reading from this paragraph. Just like us, bacterial cells can be invaded by viruses, which are small infectious agents. If a viral infection threatens a bacterial cell, the CRISPR immune system can thwart the attack by destroying the genome of the invading virus, right? The genome of the virus includes genetic material that is necessary for the virus to continue replicating, right? The genome of the virus includes the genetic material that is necessary for the virus to continue replicating. Thus, by destroying the viral genome, the CRISPR immune system protects bacteria from the ongoing viral infection. Meaning, let's say a virus attacks a bacterium. We know that viruses always take up the protein machinery of the host cell, make copies of themselves, and cause havoc in the host cell. But a way to protect themselves from viruses, a way by means of which bacteria can protect themselves from viruses is by the development of this CRISPR immune system, which is nothing but a repetitive palindromic DNA sequence, which will protect these bacteria from the ongoing viral infection. How? We will do this very soon right now. This repetitive sequence of DNA, which is found inside the bacteria themselves, will directly target the viral genome. It will break down the viral DNA and protect the bacteria from the ongoing viral replication and infection. Let's look at this simple concept map about how the CRISPR works in the body of a bacterium. And then we'll be utilizing this technique and applying it to some different situations, right? So in this diagram, I'm zooming into it. It shows you the normal immune mechanism of a bacterium. As you can see at the top right corner, a virus is invading a bacterial cell. You can see it's getting inside a bacterial cell. New spacer is derived from the virus and integrated into the CRISPR sequence. Now, what is this spacer? In this diagram, guys, you can see these square-shaped structures. You can see a green square-shaped structure, then a pink one, a purple one, an orange one, and a blue one. So what are these spacers? These spacers are actually remnants of the viral memory. For example, this bacteria must have been attacked by viruses in the past, okay? And this viral DNA was saved as a memory in these bacteria, and now this DNA is known as a spacer DNA. How will this spacer help the bacterium in developing the immune response? The spacer DNA will remain inside the bacterium, right? So that when this bacteria, the same bacterium, is attacked by the virus a second time, okay, the bacteria will have a viral memory and it will know how to get rid of this attacking virus for the second time. So this kind of a viral DNA which the bacteria has saved in its memory is known as a spacer DNA, right? You can also see the black diamond-shaped structures. What are those black diamond-shaped structures? Let me read this and I think it will be more easy for you. In this diagram, the steps of CRISPR-mediated immunity have been shown to you. CRISPRs are a region in the bacterial genome that help defend against invading viruses. These regions are composed of short DNA repeats, black diamonds, and spacers, colored boxes. So, अभी मैंने आपको क्या कहा कि CRISPR is a repetitive palindromic DNA sequence inside a bacterium, which protects it from attack by an invading virus. What is it made up of? It is made up of these black diamonds that you can see in the picture and the colored boxes. What are the black diamonds representing? The black diamonds are representing the DNA repeats, and the spacers are representing, you will see what they are representing. I'm reading again from the description. When a previously unseen virus infects a bacterium, a new spacer derived from the virus is incorporated amongst existing spacers. The CRISPR sequence is transcribed and processed to generate short CRISPR RNA molecules. The CRISPR RNA associates with and guides the bacterial molecular machinery to a matching target sequence in the invading virus. The molecular machinery cuts up and destroys the invading viral genome. Yani, K, you are shown over here some spacer DNA of different colors, which means a pink color, purple color, orange, and blue color. This shows that this bacteria was attacked by 4 different kinds of viruses. Why? Because 4 different colors of spacer DNA has been stored in the memory of this bacterial genome. Now, this time, the virus that is attacking has a new spacer DNA, which is green in color, the green block that you can see. So now what happens is that this virus is attacking, the new virus is attacking, and when this attacks, this new spacer, which is derived from this new virus, will be incorporated into the existing spacers. Done? This is step 1. Then after the new spacer is incorporated into the existing spacers, the CRISPR sequence, which you can see over here, this is the CRISPR sequence, is transcribed and processed. The CRISPR sequence is transcribed and processed to generate short CRISPR RNA molecules. The CRISPR sequence is transcribed and processed to generate short CRISPR RNA molecules. In step 3, you can see how this short CRISPR RNA molecule has been transcribed and made. Now, what will this short CRISPR RNA molecule do? It will guide the molecular machinery, as you can see in this concept map. Just see where step 3, on step 3, after step 3, there is an arrow which is going towards step 4. So just look at this part of the diagram very carefully, that after the CRISPR RNA is formed or transcribed, this CRISPR RNA will guide the molecular machinery of the bacterium to target and destroy the viral genome. Exactly, to target and destroy the viral genome. Why? Because when this CRISPR RNA is formed, it already has this new spacer which has been derived from the virus. It has this new spacer because this new spacer, or in other words, the new viral DNA, has been integrated into the CRISPR sequence. And now with the help of this integrated spacer DNA into this CRISPR RNA which has been formed, the CRISPR RNA will guide the molecular machinery to target and destroy the viral genome, right? It will now easily kill the virus and it will not allow the virus to replicate. And in this manner, you can see how the specific viral genome was targeted by the CRISPR RNA and it was gotten rid of. I hope this is making sense and I tried my best to explain it to you. Now, CRISPR immune system, of course, one of the uses is that it works in a bacterium to protect it from repetitive attacks by viral infections, right? How does it do? It does it in 3 steps. Let me repeat these 3 steps once again. Step number 1 is known as adaptation, in which the DNA from an invading virus is processed into those short segments or spacers, which will be integrated into the CRISPR sequence. Step 1. Step 2, production of a transcript of CRISPR RNA. This CRISPR RNA will be transcribed. It contains the repeats and spacers, of course. So this will be transcribed. Okay. The process of copying DNA into RNA will happen, right? Because what are we doing? हमारे पास है क्या? हमारे पास we have this thing. We have this CRISPR sequence, which is entirely made up of DNA. It is made up of the spacers, and it is also made up of the CRISPR repetitive DNA sequences. So what is actually happening in step 3 is that we have made an RNA from a CRISPR DNA sequence. Or in other words, we have changed DNA into an RNA, and now we are giving this RNA a new name, which is known as the CRISPR RNA, right? So we've made the CRISPR RNA, which is step 2, right? So again, I'll repeat step 2, production of CRISPR RNA. CRISPR repeats and spacers in the bacterial DNA undergo transcription. The process of copying DNA into RNA, unlike the double chain helix structure of DNA, the resultant RNA is a single chain molecule. This RNA chain is cut into short pieces called CRISPR RNAs. अब हम क्या करेंगे CRISPR RNA से? We want to target. So we will be using these CRISPR RNAs as guides, okay? So these CRISPR RNAs will guide the bacterial molecular machinery to actually destroy the viral material. Why will they guide? Because these CRISPR RNAs already have the spacer DNA, as you can see, the spacer DNA, which is green in color, incorporated into the CRISPR RNA. So this CRISPR RNA is going to guide the molecular machinery of the bacterial cell to target and to destroy finally the viral genome, which is mentioned in step number 3 over here, that the CRISPR RNAs guide the bacterial molecular machinery to destroy the viral material. Because CRISPR RNA sequences are copied from the viral DNA sequences acquired during adaptation phase, they are exact matches to the viral genome and they serve as excellent guides, right? So the beauty of the CRISPR technique is its high degree of specificity. It is a highly specific kind of a technique which is based on a normal immune mechanism which is found in a bacteria when it is attacked by viruses. But we will be using these techniques and we'll be applying them onto different fields of medicine. We can apply them in the field of industry, in the field of basic research, and in the field of medicine. For your syllabus, I think it would be best for you to just remember its application in the clinical medicine, right? How do we do it in the clinical medicine? You can see over here. We have this guide RNA, okay, which has the targeted DNA. We will be using this guide RNA to cut the human DNA. Let's say we want to cut the human DNA, we want to slice it, we want to edit it, we want to silence a gene, or we want to replace an allele. We can do various things, right? So this concept map is showing you gene silencing and editing with CRISPR. Guide RNA, which you can see at the top, is designed in such a manner which will match the DNA region of the interest. यानी कि आपको किसी का DNA region काटना है, you want to cut the specific DNA region, so you will design a guide RNA which will be completely in line with, which will completely match the DNA region which you want to cut of any person. And you will design your guide RNA according to that. So your guide RNA is designed to match the DNA region which you want to cut. What will that guide RNA do? It will direct the molecular machinery of the cell to cut both strands of the targeted DNA. It's cutting both strands of the targeted DNA. During gene silencing, the cell attempts to repair the broken DNA. This is a normal immune mechanism of our body, that if our DNA is cut, your cell will start repairing it naturally and normally. Okay? And this you will do if you want to just silence a gene. Okay? So it's written, during gene silencing, the cell attempts to repair the broken DNA, but often does so with errors that disrupt the gene, effectively silencing it. But if you actually do gene editing, then a repair template with specified change in a sequence is added to the cell and incorporated into the DNA during the repair process. The targeted DNA is now altered to carry this sequence. Okay. Now I'll show you from your textbook how this is done. Look at this diagram. This diagram shows you the, it's figure 19.10. It says gene editing using CRISPR Cas9 system. CRISPR is short form for clustered regularly interspaced short palindromic repeats. You can see the guide RNA at the top. Okay, you can see this Cas9 enzyme over here. This enzyme has active sites also. This is that target base sequence which we want to cut, right? Acha. In order to cut this sequence at this specific place only, we need a guide RNA, which is complementary to this targeted sequence, right? We want to, this is that part of the faulty allele that needs to be edited. So what do? We will use the guide RNA, which will target a specific base sequence on the DNA and it aligns it on the Cas9, which is the Cas9 enzyme, right? Now, this enzyme will just behave like a restriction enzyme and it will cut the DNA at this specific position. Okay, this Cas9 enzyme will cut this DNA at this specific position. So you can see this is the cut DNA, right? Now I can do two things. I can just cut it. And when I cut it, of course, what have I done? I have silenced the gene that was found over here, which was mutated and it was causing problems. So if I simply cut it and I don't replace it with a normal gene, then what will happen? The normal natural immune mechanisms of my body will repair the DNA, right? So DNA will be repaired by a mechanism that inserts a base sequence, any random base sequence, to silence the mutated allele. This can be done, or through CRISPR, the DNA can be repaired by a mechanism that inserts a short length of this DNA to correct the faulty allele, right? So the same thing has been mentioned over here in the description. It says the CRISPR-9 system has been developed so that the guide RNA can be made with base sequences which are complementary to the base sequence of the DNA that we are interested in. This guide RNA gives the genetic engineer a way to edit the DNA at specific places anywhere within an organism's genome. Who will cut the DNA? The DNA will be cut by Cas9, which is the enzyme, right? So after the DNA is cut by Cas9, the natural DNA repair mechanisms can repair the break either by adding one or more nucleotides that can change the base sequence, or by inserting a short length of prepared double-stranded DNA with a specific base sequence. So for CRISPR, we need two things. We need a guide RNA and we need Cas9. Cas9 is that enzyme which will behave as a restriction enzyme, which will help to cut the DNA, and the guide RNA will target a specific base sequence so that the Cas9 knows where to cut at the specific sequence. Now, after you've cut it, either you simply leave it so that the DNA repair mechanisms can insert any nucleotide base sequence to silence the allele, or you can also repair, DNA can be repaired by a mechanism that inserts a short length of DNA to correct the faulty allele, right? So DNA repair can take place by inserting simply a nucleotide sequence to silence the allele, or DNA can be repaired by inserting a short length of actually DNA to correct the faulty allele. Now, the applications of CRISPR can be endless, and some of the applications are described here in your textbook, so I'll be reading through them. It is possible to sequence the genome of individual people through CRISPR technology. This means that it is possible to prepare customized drugs to treat cancers. There is also the possibility of editing genomes of insect vectors to prevent the transmission of diseases like malaria, and to provide bacteria used in industry, just like we need Thermophilus aquaticus to do PCR. So to provide bacteria used in industry with protection against viruses, genetic engineering can now edit or correct the genome of any cell and organism in ways that are easy, fast, cheap, and highly accurate, right? Tens of thousands of such guide RNA sequences have already been created and they are available to the research community. CRISPR or Cas9 can be used to target multiple genes simultaneously, which is another advantage that sets it apart from other gene editing tools, right? Okay, so guys, I will now show you this very short video in which CRISPR has been explained in a very simplistic manner. So just listen what he's saying. Listen to what he's saying.
We have known genetic mutations that underlie disease. And so one of the thoughts is, if we know what mutations cause disease, why not just go in and replace it and revert it back to the normal sequence? And CRISPR is a technology that we harness from bacterial cells to be able to make those precise changes in the human genome. And the way it works is, imagine if you are writing a novel and you are proofreading the novel and there are some typos you want to fix. And let's say the sentence in the novel said twinkle, twinkle, big star, but you really want to fix it so that it's twinkle, twinkle, little star. There are different ways to fix it. You can either get rid of the word big, that would read twinkle, twinkle, star. It doesn't capture the full meaning of what you are intending the sentence to be. And other ways is to just add in the little word, so you have twinkle, twinkle, big, little star. And that still is quite confusing. So what you have to do is you have to go in and replace big with little. And so one way to do this, you may give Cas9 a RNA that encodes twinkle, twinkle, big star. And by taking this search string, Cas9 can go into the genome and try to find a place where the mutation matches twinkle, twinkle, big star. And then Cas9 would make a cut in the word big. And then by providing the cell with a repair template that carries the word little, the cell will repair this DNA break with the new repair sequence.
Okay. Appreciate it. Thank you.
Thank you.
Thank you so much.
Thank you.
Okay, so guys, I hope this was helpful. But still, if you haven't understood, you can post your questions on the classroom.